Hey everyone, welcome to this session. We're competing against ice cream. And that is really tough, but it's a shame because this is going to be a really informative session given by Dr. Cody, who's going to share a little bit about her experience with the Chromosome 18 registry. And I think there's going to be a lot of valuable lessons that she's going to be able to teach us. So I appreciate her time. With that, I'll pass it over to her. Thank you.
You need your phone here. All right. Thank you so much for eating your ice cream quickly. I appreciate that. First, let's see here. I'll just sort of introduce myself. I wear a couple of hats. I am the founder and president of the Chromosome 18 Registry and Research Society, a parent support group, but I'm also a professor in the Department of Pediatrics at the University of Texas Health at San Antonio, and director of the Chromosome 18 Clinical Research Center. So I will explain how those things came to be here. First of all, I'd like to show you my family. I have two daughters, Catherine and Elizabeth. Elizabeth is in the middle there. She's 36. She has a deletion of 18q, so she's missing 30 genes off the end of the long arm of chromosome 18. And she's hearing impaired. She's growth hormone deficient, and let's see, she has a driver's license. She got 32 hours of college credit before she decided it was just not her thing. So she has some wisdom there. She also has a driver's license, but had the wisdom to realize she is a scary, rotten driver. So she hasn't driven in 10 years. And my other daughter, Catherine, my older daughter, Catherine, is shown here with her husband and her three most amazing grandchildren in the entire universe. So this is us, and their dad passed away many years ago. That's why he's not in this picture. So I want to start out with talking a little bit about where we are now, and then to back up and explain how we got there and some of the decisions we made about how we're organized and how we got to this. So as I said, I'm president of the Chromosome 18 Registry and Research Society, and our mission is to help individuals with chromosome 18 abnormalities overcome the obstacles they face so they might lead healthy and happy lives. Sound familiar? You know, that's kind of all of our mission statements. Oops, I'm not. There we go. And the chromosome 18 group includes anyone with any chromosome 18 abnormality, whether it's a duplication, a rearrangement, or a deletion. And we started this way because we were told when Elizabeth was diagnosed 36 years ago that there were 65 families in the world. So I thought, okay, if we include all chromosome 18, we might get 100 families. And so the group, the support group, lumps everybody into these 5 groups. So people with 18q deletions, and let's see, do I point it here? Where do I point this to? Maybe I said I wasn't going to use the pointer. That's okay. If you have an 18q deletion, people have a deletion of any size from any portion of the long arm of chromosome 18, and they have 18q minus, shown here on the left. Likewise, there's 18p minus, people with deletions of the short arm, again, any size, any location on the p arm. There are people with ring chromosome, people with an isochromosome, that's the most rare tetrasomy 18p, so they have 4 copies of the p arm. The most common by far is trisomy 18, except that those individuals tend to be very medically fragile. 90% die before their first birthday, and there is already an organization for those families, so we really haven't focused on that, but we of course never prevent anyone from joining our group. But we always make sure that any trisomy family that contacts us knows about the trisomy support group. So those families have a very different set of needs than our families. We currently, it's a little more than 100 now. We have about 4,000 members worldwide, and we have a European and Australian organizations as well. We have an annual, not pre-COVID, have an annual conference. Every 4th year it's in San Antonio. The Europeans have or had one every other year, and Australians every 4th year. And of course, we do all the things that support groups do. We have newsletters and Facebook pages and website. We have 3 full-time employees, an executive director, a program director, and an operations director, and then a part-time bookkeeper. And this is ingenious for our executive director. We have this huge bevy of work study participants from all the local colleges. So at any one time, we have about 12 work study people who are, they're college students, they're getting paid, so they got to show up, they're not volunteers. And they actually do lots of the social media kind of stuff that that age group is so good with. And help with the newsletter and all kinds of activities. So they've really been invaluable for free labor. Also, we have the Chromosome 18 Clinical Research Center at UT Health in San Antonio, as I said, in the Department of Pediatrics. And our group of investigators is large. We have, as you can see, pretty much every medical specialty. Let's see, psychiatry, psychology, neurology, endocrinology, ENT, ophthalmology, you name it. And for any one family, any one of these specialties could be their sort of main go-to specialty. It kind of depends on the child. So we have to have this huge group. And as we've learned more, for example, that people with tetrasomy 18p have bone mineral density issues, we luckily, right around the corner from my lab, had someone who specialized in developmental bone density. And so he's come on board, and when we learned there was a gene that might be involved in abnormal platelet function, right across the hall from my office was someone who specialized in platelet function. So we've brought on more specialized expertise as we've learned more. And then, of course, there's the basic science side as we've learned which genes to focus on. People involved in mouse behavior, mouse model development, CRISPR techniques, iPS cells, and mouse electrophysiology, that sort of thing. So we've got a pretty big team that would really be hard to emulate at lots of other places. In supporting that team, we have a staff. Our administration people are really supported by the department. The other people are supported by the research center. One person who runs the lab operation, and because most of the basic science stuff is collaboration, so she helps facilitate all that and enroll members, or enroll participants, sorry. And then, of course, a research team with a psychometrist and genetic counselor and research associates. We have almost, probably by today, we probably have 700 families enrolled, and you can see on the research side, we have more categories we divide people up into, but we have almost 700 people enrolled in the research study, which is different from the support group. And they come from all over the world. The limiting factor is really the ability to get blood to us within about 3 days, because one of the things we do is establish cell lines from the blood sample, as well as having DNA in a cell bank. We collect medical records, and this is ongoing. We bug people continuously. You have a new finding, we need to see those records. We put them in our database. We do lots of surveys. Sometimes there are surveys about, how many broken bones have they had, or what's their current living arrangement, those sort of things. But sometimes they're standardized neuropsych surveys that can be mail-out surveys. All that goes into a database. Of course, we do the DNA analysis, so we have high-resolution breakpoint data on everybody in the cohort. We do clinical evaluations. Right now, we don't have funding for that, but we've done many, many evaluations where people come to San Antonio for a week, and they see that whole list of specialists within a week. And of course, the point of all of this is then to turn it around and provide information for families. So how we got there really began here. This is in 1985, so it's been a while. These are my kids in 1985, and Elizabeth, my younger daughter, who you already met, was born with a bilateral cleft palate and cleft lip and metatarsus adductus, so her feet turned in. But, she was bright-eyed, she was alert, she was eating okay, amazingly eating okay, given that her mouth went from here to up here, and no roof to her mouth at all. So when the doctor called me in to pick up some test results and said she has this, she had a deletion of the end of chromosome 18, this is me, and this was you, you can relate. What the heck is this? What does this mean? Is she gonna live? Am I gonna live? Where do we go? Who do we ask? What do we do next? And this is kind of the information we had. A book with really awful pictures that said things like, well, they have big ears and too many whorl patterns on their fingers. And, nothing about management, nothing about outcome, and other than mental deficiency and obnoxious personality. I've yet to meet one of those, though. The geneticist said, I guess we'll just have to wait and see, which is truly unsatisfactory. And so we were very lucky. My husband was in the Air Force. And we were recently, when she was 6 weeks old, stationed in San Antonio, where the Air Force has their largest training hospital. So we pretty much had unlimited medical care. Never had to fill out a form or pay a copay or anything. I just had to talk somebody into doing an assessment, which wasn't always easy. I got really good at that. But by the time she was 5, after being told multiple times that, sorry, they're all short, I finally got 6 months worth of assessments done, only to find out she's growth hormone deficient, which is a treatable condition. And when treated, she started growing like crazy, and she got her muscle tone improved, and she just seemed more alert and together. It made a huge difference in her life. But when she was 5, we learned she was hearing impaired. We didn't know that. Her ears worked. It was the connection between her ear and her brain that didn't work so well. So she heard garble. She repeated garble. She repeated what she heard. And we looked at her like, huh? And boy, did she think we were stupid, because we couldn't understand, even though she's repeating just what she heard. So she was one frustrated little girl. And at this point I said, I got to find those 65 other families. So with the help of her neurologist, we started the Chromosome 18 Registry and Research Society. And I'm going to this is a little tangential pictures of the house. So do you have a question? Oh, okay, you're fine. And if anybody has a question, please raise your hand. So our initial challenges, which you may have or you may not have in your organization, was that we couldn't find any experts out there. The only people who knew about these conditions were lab directors who don't know squat about clinical management, much less any of the physical manifestations people have with these. Geneticists, or I hope there's none in this room, they're diagnosticians. They don't know anything about management. They should, but they don't. So we couldn't find anybody who really knew anything about these conditions. So we thought, okay, we're going to do surveys ourselves. We're just going to collect this information. So we do a survey and we'd ask questions. We'd ask for medical records. And then we would get information back that maybe had 2 different big words having to do with, let's say, hearing. And we didn't know, are these synonymous words? Is one a bigger, worse condition than the other? Is one a subset? Is one a precursor to the other? Well, we didn't know. We needed some experts, but there weren't any out there. So this was our major problem. And the other big problem is we had no control. So what happened was a couple of things. One of our family members was contacted by somebody who wanted to investigate these conditions. We're all excited, yay, she gave blood, and she then gave me the consent form. The consent form had nothing to do with a chromosome 18 abnormality. I don't know what this person wanted her blood for, but they got it. Never heard from them after that. Somebody else wanted to understand a neurologic condition that one of their patients had who had 18p minus. And so they wanted to enroll other families. So we told the families about this person who wanted to do this survey and ask them about what they'd experienced if they'd had this neurologic condition. So we told our families, they contacted them. And then a few months later, families are calling me all mad because they're saying, now this guy wants blood. You never told us he wanted blood. He didn't tell me he wanted blood. And then another neurologist comes along with really a better question, but our families had already participated with that first guy, so they're not going to participate with the second guy. So that's when we said, okay, forget this. We have to take charge of this. We have to direct this process. We can't have families just participating in the first study that comes along. We need to orchestrate this and take control. So those were our major challenges. So what did I do? So I enrolled in graduate school. If you can't find somebody to do it, you just have to do it yourself. And so that was 1991. I finished in 97 and immediately really started on this project to really at first just collect blood samples. And my PhD project was really to do the molecular analysis, which, in those days, the markers to do that were just being published. And so as soon as some were published, we employed those. So it was a very new technology in those days. And also really began building that team just immediately when I started graduate school. And most of those people are in the Department of Pediatrics. And I was lucky enough that my lab mentor had a cross appointment to pediatrics. So that's really where I hung out all the time. And I actually did all my work in a lab of one of the pediatrics faculty who didn't really have a lab project. So that's been my home this entire time. And we have had an absolutely laser focus on those questions that are going to help us eliminate obstacles to independence. Maybe that's an unrealistic goal, but at the same time, if it's not your goal, you're never going to get there. So we haven't really worried about causes of chromosome 8 of abnormalities, because that's not really going to matter. It might be NIH fundable, but it isn't really going to help our kids. So we've not focused on that. We're really focusing on those things that are going to help families. And so at this point, I want you to forget everything you've ever learned about how one approaches rare disease research, because this isn't like any other rare disease research. This is entirely different. We are a different boat altogether. So forget all that. So these are the things that make us completely unique in the rare disease world, which the higher ups in the rare disease community, in spite of me harassing them for 20 years, haven't quite appreciated. First, these conditions, as you know, are genotype defined. So anyone with any deletion of 18q has 18q minus. It could be a little deletion at the end or a little deletion way up in the middle of the long arm. They're still 18q minus. It doesn't matter whether they do or don't have hearing impairment or seizures or whatever. None of that matters. They all have 18q minus if they have a deletion of 18q. And because they're defined genotypically, they may have heart problems or bone problems or cognitive problems or eye problems. They could have any range of problems. There's not a single specialist that, that focuses on the issues that are important to our families. So also, they're ultra rare. In the case of the chromosome 18 conditions, without trisomy 18, all the others combined occur in 1 in 24,000 births. So if this room were filled with pediatricians, maybe only 1 or 2 of them would ever expect to ever see someone with a chromosome 18 abnormality of any sort in their practice. So they're ultra rare, which means you have to think about how you organize your research and your families in a very sort of different way. Also, as is obvious, I hope at this point, there's a huge variety in the size and location of the gene or the chromosome change. There's lots of heterogeneity within these groups. In fact, no 2 people with an 18q deletion have exactly the same deletion. And as you saw, we have about 400 of them in our research that we've assessed so far. No 2 people with ring 18 have the same exact deletion. With 18p minus, half of them are missing the whole short arm. The other half have something unique. Tetrasomy 18p is almost all the same, but across all these, there's so much variability from person to person that you can't just do a syndrome description that really fits everybody. Also, of course, there's genetic complexity, which is obvious, I think, to all of you. There's all these genes. There's 263 genes on chromosome 18. We don't know what they do. There's all these phenotypes, these conditions that might affect the heart or the brain or the eyes, and we don't know which genes are impacting those things. So we have this huge genetic complexity going on. I'm going to skip those. And then, of course, there's outcome complexity. So that even when we figure out maybe some of the key genes, what we're going to find is that one organ might be affected by two genes. There may be two genes required to cause one problem or two separate problems in the same organ. There might be one gene that affects two different organs, or there might be one organ that's affected by many, many genes. And so sorting out the manifestations, the characteristics, the phenotypes, whatever you want to call them, is really going to be a challenge, and linking those to specific gene action. So this is where we are, but not to stress out too much because, you know, it's just biology. If it's biology, we can figure it out. Doesn't mean it's simple, but it's biology, it's science. We can get there and we can figure it out. So how have we addressed each one of these challenges? Well, first, of course, they're genotype defined, and that's by definition that we can't change. It's ultra rare, so we have created a single coordinating center where all the data is going to reside from everyone. And because there's so much heterogeneity between different people with these conditions, you can't really do a syndrome description. So our plan is to instead think about the specific genes and the role they play. So any one person could learn that, okay, you have a deletion of 8 genes. Well, these 6, they don't really play a role. This one puts you at risk for something, so we'll monitor that. This gene causes this problem, and this is how you manage it. So we could make, if we organize this and think about these conditions as gene by gene, then we have the ability to make personalized, individualized, appropriate management or syndrome descriptions for each individual person. Also, the genetic complexity, the way we are addressing that is to try to identify the dosage sensitive genes. Really, only 10% of genes are going to be the ones that impact a person when you have too many or too few copies. So we've really focused on reducing or understanding the genetic complexity by trying to identify those few key genes. And then the outcome complexity or the phenotype complexity is by doing lots of really extensive characterization. And by that, I don't mean just every body system. I mean, as you figure out first that they're all short, then you ask why they're all short. You learn they're growth hormone deficient, then you ask, is it hypothalamic or pituitary in origin? So you're always fine-tuning the phenotype and the clinical assessment so that you can get closer and closer to the physiology or the gene action. So those are the things that define our approach. And so, as I've already said, the approach is to have a single clinical research center. And this also means that families just have a single point of contact. They want to, a researcher wants to study some aspect, they contact me. We see ourselves as a coordinating center. We have lots of collaborators. But the families don't ever have to worry about those other people. They always have one place to go to ask questions. And as I said, we do a comprehensive clinical evaluation and collect all aspect of clinical and developmental assessments across their lifespan. This means we, of course, have a more consistent set of clinical data, really important, especially for us for MRI scans and for the neuropsych assessments. And because this is ultra rare, then we have one center with a critical mass of patient information. And you don't have to try to coordinate people at this place and people at that place and try to get them to do all the same things and really share their data like they should, and and get them all together and get them on the same page. Everybody's in the same place. And as I said, this isn't one medical specialty. This is this whole group of medical specialties. It's really hard to duplicate in lots of places. And if we had 4 or 5 research centers around the country, 4 or 5 families wouldn't have to travel. Everybody else will still have to travel. So it doesn't really, in the convenience side, it really doesn't make much difference. But also being at a university, we have the longevity. It's not a company. It's not going to get bought out. It's not going to, go under. It's going to live. The UT system is not going away anytime soon. These resources and this project can evolve as as faculty members retire and new ones come on. So we have the ability this has to go on long after I'm gone. And so being at a university helps ensure that. And also, this is a lifetime longitudinal study. So we're collecting information about people as they get older, because we have no idea what, are they at high risk for Parkinson's or Alzheimer's? Nobody knows. Do they die early from something we could prevent? If we knew about it, we don't know. So this is as I said, a lifetime longitudinal study. Now, just to be clear, as I said, I wear 2 hats. There's 2 organizations. The Chromosome 18 Registry is the advocacy group. So like this organization, we hold conferences, do member communication, lobbying or educating policymakers and the public, and fundraising. So the Chromosome 18 Registry raises money. The research center spends the money. And everything at the research center, of course, has IRB approval, and so you have to sign consent forms. We do the bench research, or we coordinate with people who do, or collaborate with people who do, likewise with the clinical research. And we have expert collaborators really all over the world who are experts in specific genes that we're interested in. And of course, data analysis is centered at our institution, but we have the ability to share de-identified data with people who can help with analysis. What we don't do is share names. New family joins the Chromosome 18 Registry. The registry never ever gives the name to the research center and says, this is a new family, contact them. Never happens. The reverse never happens. Families have to enroll with one and the other, or neither, or whatever they want to do. It's up to them. It's their deal. No pressure either way. So our general research strategy really has 2 parallel sort of tracks. One is about people and one is about genes. So we're, of course, investigating the clinical features, which I guess you figured out by now. But what that results in is defining standards of care. So what kind of assessments need to be done? What conditions are more common? What do you need to look for? It gives families something to give to their physicians who aren't going to ever see another patient like this, some guidance. As I said, we're interested in the natural history, what happens to affected individuals as they get older. We're also interested in biomarkers. So as we try to understand the more physiologic nature of the conditions, what physiology or other biochemistry or other things that might be potential clinical trial endpoints. So we're trying to set ourselves up for future clinical trials. And because these are so rare, we've really started to focus on remote assessment. So we're trialing different devices that are kind of like a Fitbit or iWatch that actually let you download the raw data, which is the hard part. So we're trialing different ways to assess multiple biological parameters in that way, in a remote way. Not to mention, of course, just plain old surveys over the internet, some of which we make up questions to understand a general area, and some are very specific neuropsych assessments. And then we're also trying to identify the key genes. Like I said, really only 10% of the genes are going to be the genes that are dosage sensitive, that make a difference when you have 1 copy or 3 copies or 4 copies. And defining the action of those genes in cell lines, in iPS cells, in mouse models, and then learning to regulate that gene action as a way of correction for future therapies. So we see both of those parallel tracks. I mean, I think the people side are the immediate answers, and the gene side are the long-term answers. Just as an example, which I've sort of already said, for example, in the people we were told early on, they're all short, and we learned they could be growth hormone deficient if they have an 18q deletion. Actually, we now know exactly which region of the chromosome is responsible. We don't know which gene, but we know down to a few genes, and that they're eligible for growth hormone treatment, which it makes a big difference, a positive difference. Likewise, on the gene side of the equation, there's a gene on 18p called SMCHD1. And it was identified as being when 1 copy has a mutation that causes it to not work. So a loss of function mutation in 1 copy, so kind of like having a missing copy, people get facioscapulohumeral dystrophy type 2. Took me a long time to learn how to spell that. And so we worked with a group in the Netherlands who specialized in that, who helped us assess the genetics of our population. It's a really complicated genetic underpinnings of this. We have a few people we think might be at risk, but it's adult onset type of muscular dystrophy. So we continue to assess everyone with an 18p deletion for first signs of this condition. Luckily, some of that we can do remotely with a video interview, so we're collecting those. So some of our findings come from assessing people and some from what we learn about genes and gene action. So these are just 2 examples. And we realized that physicians of our families, they're not looking up any of our papers. That's just, it's not presented in a way that makes sense. And there might be one on growth hormone deficiency and another one on seizures, and they don't have time. And so we reorganized all that information into what we call physician management guides. The terms aren't explained because the guide is for physicians. The version with explained terms and all those, that's on the advocacy group website. These are on the Research Center website. There's one for each one of the conditions. And this kind of comes in a couple flavors. They all have a 1-page, 60-second summary. So maybe an ophthalmologist could just look at that and say, oh, this child I'm going to see has tetrasomy 18p. I don't know what that is. It kind of tells them what it is, so they're not going to look too stupid. It tells them that this child is not medically fragile. They're probably ambulatory. In fact, they're probably hyperactive. So maybe, you know, that might mean to them they allow extra time. So it's just a really global 60-second summary things. And then it's followed by, in this case for 18q, 23 pages of information. And the page I'm showing you here is kind of the table of contents page. So it's organized at the beginning by the things you need to do in a neonate. Then the referrals or exams you need to do in someone when they're first diagnosed, the referrals you need to make. Then for ongoing management, all the things you need to monitor, as well as things that need to be assessed really on an annual basis, for example, thyroid conditions in this population. Then everything we know about adults with this condition. How many are married? How many live independently? How many are in group homes? How many finished high school? All that. That's the information we keep collecting from our participants. So families can see right away kind of what the adult population looks like. Even though everybody's a little different, that still is better than having nothing to know. And then last, the individuals who have passed away and their cause of death and the age, and compared to the age and the population of the whole cohort. So there's a lot of information here. Like I said, 23 pages. And one of the aspects of that I wanted to share, so we took the milestone information we got from everybody, and we made bar graphs. And so these are at the top part in the yellow and green are the milestones organized by age. The green region is the typical age that that milestone is acquired. And so the X shows where in 18q deletions, the average age that milestone is acquired. And the ball pointed line showed the entire range. So this is really also for parents. It's not standard deviations, it's the whole range. So you can see it's a really big range. And so you can see the milestones and where they're acquired and typically when they happen. Below that is just a small example that I have to say made our neuropsychologist's head explode when she saw what I did with her data. But she's going to have to get over it because I'm the boss. So I'm sure you know, you've done neuropsych surveys. There's one that's going to have a range from 1 to 20. Another one has the outcomes are from 1 to 100. Sometimes the big number's good. Sometimes it's the big number that's bad. And I can't remember all that. And so a table with all those numbers I found completely useless. So I translated every test that we do into a bar graph that it has 3 components: green for normal, typical, acceptable, whatever you want to call it, range, depending on the assessment, yellow for at risk, and red for clinically significant. And so different tests will have the green bar, in different place and different size, but they all go from green on the left to red on the right. So, and they all have the actual numbers of the scale right across the top. So you could take your kids' numbers and see where they actually fall. But again, and this is broken out by age range. This is a social responsiveness scale. So, and it's also broken out by each subscale within the test, because this is what's really important to parents, is to really understand the fine-grained aspects of what to expect and what kids might be good at or might need more emphasis on and help with. So, and this is also divided by age range. So you can see for social awareness, preschoolers are kind of the normal, edge of normal. They're in the at-risk as they get a little older. On social cognition, it's sort of the same sort of picture. And so all the subsets of subcomponents of this SRS test are shown below by age range. And we have, I don't know, half a dozen neuropsych tests all displayed this way. So you can hopefully understand the data a little bit better. But again, that's something that your neuropsychologist won't like, but that's okay. It doesn't matter. This information is really for you. It's not for them. And just to kind of summarize where we are finding key genes, I know you can't read this, and that's okay. That's not really the point. Down the left-hand side are all the phenotypes that we've associated with individual genes. And those that are bolded are phenotypes or characteristics that occur in more than 50% of a person with a deletion. This is really just for deletions. Those that aren't bolded occur in less than 50%. So you can see most of these conditions occur in less than 50%. Many of them 10% or 25% of the people with a deletion of that gene. On the right-hand side is our work to be done. Each one of those bars on the right-hand side is a region associated with a phenotype. Let's say congenital heart disease. So we've identified a region. Everyone missing with congenital heart disease has a deletion missing that region, but we don't know which gene yet or genes in that region are responsible. And again, the dark bars are things that occur in more than 50%, and the light bars are less than 50%. And so you can see in the low occurring, less frequent characteristics is a lot harder to figure out which are the key genes because fewer people have those characteristics. But that just kind of shows you a little bit of where we are in figuring out the gene by gene effect. In total, there's 263 genes on chromosome 18. And actually, we've been able to classify 239 of them. So almost all of them, there's enough information to make a first guess at their potential impact when you have a deletion or duplication, in this case, deletion. And 149 of them have no effect. So those I don't even have to worry about right off the bat. 66 are considered a risk factor. So you need a deletion of that gene plus some other genetic change to cause that condition. So really only 24 are considered causal out of 263. So again, that's less than 10%. And again, of those 24, about half of those cause a problem in less than 50% of the people with the deletion of that gene. So so we have a lot to understand. There's so much variability, but we're making a lot of progress by going gene by gene and trying to understand the effects. The obstacles, of course, are time and money. Nobody knows better than you all about that. And just to kind of share where we are and what it has taken. The Chromosome 18 registry families, this is over 30, almost 30 years, have put in about $6 million. We had very, very early on that really got this jump started. One family and their company who matched their donation, which of about $3 million. Money's long gone. And a little bit of NIH money, also a little bit of support from UT Health, and then some small grants from the foundations or individual donors. So this is the kind of money it takes to at least get started. We have a long ways to go. And it costs about $300,000 a year to maintain the infrastructure, the staff I showed you, to keep enrolling people, keep doing surveys, all the long list of specialists, those are all volunteers. Or they're on a separate grant that supports their activity, not supported by our center. So lessons learned. Really, don't forget, the technical experts, the scientists can tell you if what you want to do is technically feasible. They can tell you if it provides new information and what that information might lead to. They cannot tell you what's the most important thing to do. That's your job. And I see too many groups defer that to the scientists. They can't tell you what the most important thing is. You're the only people who can define what the most important thing is. What's the most logical thing to do and the most responsible thing to do given how much money you have. So these are the questions that you all need to be asking, are what your priorities are. What's the most important thing to do? And is this really the direction we want to go? And will this get us closer to treatments, which is really the bottom line. Everything has to be focused toward getting closer to treatment or management. Yeah. So this is something I don't think I really have to explain to this group because it's really obvious that you all get this. But to never forget that life is not about how you survive the storm, but it's how we all danced in the rain together. So thank you for your attention. I'd be happy to take any questions about anything. I'm here to tell you everything I know. If I could possibly do that, and I went way over. So anyway, thank you for your attention.